Now, let us understand the life cycle of viruses. We are going to learn this in general. So, what we understand is that from the definition of viruses, obligate intracellular parasites. That means they enter into a living cell, whether it is eukaryotic cell or prokaryotic cell, and after its entry inside the cell, it releases its genetic material or genome, which may be either RNA or DNA. And after the entry of that genetic material in the host cytoplasm, this genetic material then undergoes replication and transcription and translation. As a result of that, it leads to formation of replication of genetic material, either DNA or RNA. It leads to increase in the number of copies of that nucleic acid. Transcription and translation of that genetic material, it leads to synthesis of proteins. These proteins are nothing but the components of virion. And finally, this nucleic acid and the proteins or the components of virion, they assemble to form a complete virion, which then escapes from the cell either by the process of exocytosis or by bursting of the host cell. So, this is in general. Now, let us try and understand the life cycle of an animal virus which has caused a recent pandemic, that is COVID-19, the coronavirus. Now, this coronavirus, as we understand that it is a close relative of influenza virus. We are already seen the structure of coronavirus. The blue plate that you are seeing is the surface of cell. For example, it is alveolar epithelial cell, or it is the host cell. You can see that this host cell possesses certain chemical groups. These chemical groups helps in the binding of viruses to the cell. These chemical groups are called as receptors. So these receptors are highly specific. So this virus, the first step is binding of the virus and viral entry via membrane fusion or endocytosis. Endocytosis means the whole receptor virus complex along with the cell membrane, it is taken inside the cytoplasm. This is how the endocytosis occurs. You can see that the virus is being taken inside along with the membrane, cell membrane. And then the next step is release of viral genome. Now this coronavirus, it is a RNA containing virus. So the genetic material is RNA, single stranded RNA, which is also called as positive strand. So after the entry, this endosome, it releases that virus inside the cytoplasm, and then the protein and lipid coat of this virus, that is envelope, it gets destroyed. Whatever the capsid is there, capsid gets destroyed in the cytoplasm, and this RNA, it is released inside the cytoplasm of host cell, in this case animal cell. You can see green colored RNA, and then this RNA, once it is released into cytoplasm of animal cell, it is bound by ribosomes. Ribosomes, as we know, these are the sites for the synthesis of protein. So that is the second step, release of viral genome inside the cytoplasm. Then the third step is translation of viral polymerase protein. So you can see in upper second step, after the release of virion, ribosomes binds to that RNA. Then it leads to formation of mRNA. From a part of this RNA, it gets translated. So that result of translation is synthesis of this third step, that is viral RNA, viral polymerase. So this viral polymerase, it is a protein which is a result of first step translation. So this is viruses own polymerase. So that is the third step. So a small part of RNA, it undergoes translation. It leads to formation of viral polymerase protein, which is an enzyme. Then the fourth step, once this polymerase is synthesized, this polymerase will help this RNA, that is positive sense RNA, to undergo replication. So the function of this polymerase is to form multiple copies of RNA. That means it helps in replication. It performs replication. Replication means to make exact copies of itself. So this is how this RNA polymerase, it synthesizes several strands of RNA. That means multiple copies of genome, viral genome are synthesized. We are not going into the too much detail. This is the 4A step which shows the synthesis of RNA. Multiple copies of RNA are being synthesized. Here you can see the multiple copies of RNA are synthesized. Then the next step is these RNAs then undergo the process of translation. Fifth step. And as a result of translation, you can see translation results into the synthesis of proteins. What are these proteins? These proteins are nothing but the components of that virion. For example, nucleocapsids, spikes, membrane proteins, and envelope proteins. So in fourth step, there is replication of RNA. In fifth step, there is synthesis of viral components. After the synthesis of viral components, the next step would be after translation. You can see as a result of translation, viral structural proteins are synthesized. Then the next step is assembly. You can see that N protein, that is capsid protein, it binds around the nuclear material, that is RNA, to form nucleocapsid. So viral genome and these proteins, they combine to form a nucleocapsid. That means this is the step of assembly. Now all the viral components are synthesized and the RNA genetic material is synthesized. So these are the steps that shows the assembly process. Combination of nucleocapsid and other proteins which forms the envelope. And the final result of assembly is a complete mature virion. This complete mature virion is formed in the Golgi bodies or Golgi vesicles. So you can see that it is synthesized in Golgi vesicle, which is the normal function of Golgi bodies, that is protein targeting. So once the proteins are synthesized, they are targeted with the help of Golgi bodies. So here the virion takes the advantage of function of that Golgi bodies. And this is how the synthesis of complete virion particle is complete. This is, in this picture we are showing only the synthesis of a single virion. However, this process is 1000 times repeated so as to form thousands of viruses in a single host cell. After the assembly synthesis, now the virion is ready to exit the cell. And for leaving the host cell, again the process is exocytosis. So through these Golgi bodies, these viruses are released out through the process of exocytosis, which is also a natural process of animal cell. And this is how the viruses are released outside the cell. And these viruses are now ready to infect new cells. And this is how the infection progresses. And this process is very, very fast. This is in general regarding the life cycle of animal virus, for example, coronavirus. Similar events happen in case of life cycle of most of the viruses, whether they are plant viruses or animal viruses. Now, another example to explain the life cycle of viruses that belongs to a different category. This category is bacteriophage, life cycle of bacteriophage. Bacteriophages, as we have already learned, bacteriophages are the viruses of bacteria. So these viruses infect bacteria and leads to either the killing of bacterial cell, or it may leads to a dormant period that we'll learn in next few slides. So the life cycle of bacteriophage, it begins with, so here I am showing you the life cycle of T even phage. So in this picture you can see the life cycle of T even phage. The bacteriophage is attached to the cell wall of bacterium. So here in this case the host cell is a bacterial cell. So the first step is attachment. The phage attaches to the surface of the host cell. Second step. Then we have seen the structure of this bacteriophage, that it contains icosahedral capsid, double-stranded DNA present inside it. Then there is a contractile sheet which acts as a syringe. So, and it contains tail fibers, tail pins. So binding of phage on the cell wall of bacterial cell is possible through tail pins. So these tail pins gets inserted on the cell wall of bacterium, which is aided by the tail fibers. So the function of uh, tail pins and tail fibers, that they help in the firm binding of the phages onto the cell wall. This movement is just similar to the movement of while mosquito sits on our skin. So this mosquito first sits on the skin. That means bacteriophages, with the help of tail fibers, they sit on the cell wall of bacterium. Then in case of this mosquito, then bows down, तो थोड़ा खाली येतो, and then inserts its antenna into the skin. So similar event happens here. This bacteriophage then comes down and inserts its tail pins onto the cell wall. So that is the firm binding. Next, this contractile sheath, it gets contracted, and contraction of this contractile sheath, it leads to the injection of DNA which is present in capsid inside the cytoplasm of bacterium. So this event is just similar to the syringe action. So this is how the DNA is injected, phage DNA is injected into the bacterial cell cytoplasm. That is the second step, penetration. The viral DNA enters in the host cell. Third step. Now here all the steps have been combined. So you can see that in second step only viral DNA enters inside the bacterial cell cytoplasm. The rest of the component, that is capsid, contractile sheet, tail pins, tail fibers, remains outside the bacterial cell, which ultimately fall off. Then the third step is biosynthesis. So this DNA which has entered inside the bacterial cell undergoes transcription, that is synthesis of mRNA. So for this synthesis, bacteria's RNA polymerase is used. Then after transcription, it undergoes translation. Translation leads to the synthesis of structural components of this bacteriophage, that is, the proteins required for synthesis of capsid, proteins required for construction of contractile sheath, base plate, and tail fibers. That means translation leads to the synthesis of components of bacteriophage. Along with that, this phage DNA, it undergoes replication, and for replication it makes use of bacteria's DNA polymerase enzyme. So this means that this phage makes use of host machinery, metabolic machinery, for the synthesis of its own components. Own components means the phage genome, that is DNA, and structural components of the phage. So in this picture you can see biosynthesis part. So it involves replication of DNA, transcription and translation, which leads to the synthesis of phage proteins. Then the next step, which is not shown, which is shown in this itself, it should be the next step, assembly. Assembly means whatever the genetic material, DNA, phage DNA, which is synthesized, and all the structural components, they start assembling. So the next step is assembly. As a result of assembly, the complete virion particle is synthesized. The step is called as maturation. You can see now inside the cell a complete virions have been synthesized inside the bacterial cell. So new phage particles are assembled. And finally, after the synthesis of complete virions, these virions are released out as a result of thousands of viruses, bacteriophages inside a single bacterial cell. It leads to the bursting of cell or lysis of cell. As a result of lysis, viruses are released outside of the cell, and these viruses, bacteriophages, are ready to infect a new bacterium. This is called as the lytic cycle of bacteriophage, which happens in case of lytic phages that infects bacterial cell.